thank you very much. It's really my pleasure to share with this audience. I really don't have a background in veterinary science, so bear with me, but I think I'd like to bring you a bit deeper in the cells of your beloved animals, but also in the cells of human bodies, and tell you how we look at translational science to bring the new nutritional innovations of tomorrow. And this really requires deep understanding of the mechanisms of aging, of sarcopenia, and this is what I want to tell you about today. So as you just heard, I'm an employee of Nestlé Research. Our headquarters are based in Lausanne, where we have 2 sites, and actually I'm leading one of the sites on the PFL campus. So right next door to the university, we have student programs. We have students sitting in our labs who study in the university and run the PhDs with us. And to start with, I want to a bit go back to the basics of the skeletal muscle tissue and tell you a bit more about this favorite tissue that we study. And muscle is extremely plastic. As you can see, adaptations occur both in health and disease, adaptation to the environment, adaptation to the nutrition, but also to the amount and the type of training in healthy environment. And of course, the disease pathologies also impair directly both the amount of muscle, but most importantly, the contractile function, as well as the metabolic properties. And to deep dive a bit into the structure, it's quite fundamental to really understand the different elements of the muscle fiber structures to then derive concepts for intervention. And if you look at a muscle cell, it's actually one of the rare cells of the body that's multinucleated. If you look at the cross-section, the muscle fiber has multiple copies of the genome, and the rest of the fiber in this black cross-section here is actually full of contractile protein for contraction and for locomotion. And this is quite key because the amount of proteins that are consumed in the diet is directly linked to the ability to really store protein and generate contractile machineries. But of course, contraction is not only about the amount of proteins in the fibers. There's also a much wider contribution. And muscle mass is really controlled by both genetics and lifestyle. Actually, animals have taught us a lot about the genetics, much more than humans. And one of the most famous pathways is this TGF-beta related pathway of a soluble molecule called myostatin. And mouse studies have identified this gene as the central regulator of muscle mass. And by retrospect genetic analysis, we realized that a lot of the breeding, both in agricultural science, but also in dog racing, actually led to genetic variations in the myostatin gene. And you can see examples both from bulls, from whippet dogs, where natural selection has led to mutations in this myostatin gene, and the loss of function mutant gives hypermuscular phenotypes. What's quite interesting is actually in racing dogs, the heterozygous mutation gives a running advantage. So a lot of these athlete whippet runners actually are heterozygous for myostatin. And once in a while it happens that you get these very hypermuscular that of course is better at keeping your backyard than running a race. And this is when the mutation leads to a homozygous mutation of myostatin. The environment is also key, especially the activity training. It's very well known also, like in the cyclist sports field, the type of adaptation you do will lead to the type of muscular growth and hypertrophy. But the contractile function is not alone, and for muscle to work efficiently, you really need different cell types, different cellular structures, and different subcellular mechanisms. And the first layer comes from the structure itself. You see these long, elongated muscle fibers full of contractile protein. They have to be vascularized, of course, and the supply of both oxygen and nutrient is key for contraction. And then they can be plastic and repaired. So you have a small cell type that's very quiescent, that's called muscle stem cell or satellite cells, which I'll describe later in the talk, which actually are the firefighters of muscle. They repair the damaged fiber after an exercise. And then the fibers themselves come in different flavors. You've got different type of metabolic and contractile properties according to the fiber type. And finally, there's two other structures that are absolutely key. The neuromuscular junction that you see has this pretzel shape that transmits the voluntary signals from the brain via an acetylcholine transmission at the synapse. And this then leads to excitation-contraction coupling and to the sliding of the actin and myosin filaments. And finally, last but not least, you'll hear a lot in my talk, the mitochondria, these little powerhouses in our cells that really use up the nutrients, turn them into ATP for energy generation. They're located both at the periphery of the fiber and in between each of the Z-discs, these contractile units, to generate directly the energy for contraction. Now, you've heard a lot about sarcopenia this morning, and we fully share those definitions. For us, sarcopenia is really the age-related loss of muscle mass and function that's really core to aging. It's not muscle wasting that's caused by a disease, which is cachexia. It's different than a dystrophy that's caused by a genetic mutation, but it's really this chronic decline that happens during aging. And this curve is actually the average curve, but what happens is really high heterogeneity in the rate of decline. Peak muscle mass will also to really control the lifelong reservoir of muscle contractile apparatus, but the rate of decline is extremely variable, and we're very interested to understand the factors that eliminate really the healthy aging, this natural physiological decline that leads to decreased performance, but that doesn't lead to loss of independence, versus really the pathological aging of muscle, which we call sarcopenia. And this notion is quite important because in humans, sarcopenia has been recognized with an official disease code a couple years ago. And it's really about two main factors. It's losing muscle mass on the one hand and losing muscle strength. And what we've realized over the years is these two factors are not completely overlapping. They're related, but they certainly are governed by different physiopathological mechanisms. And one of the challenges, of course, like many chronic diseases, sarcopenia is extremely multifactorial. There's, as you can see on this slide, many causes that range from the environment, the lifestyle, the type of nutrition, but also chronic events like accidents or injuries, physiological perturbation, the metabolic fluxes, the ability of hormones and nutrients to stimulate anabolism. And then there's local tissue challenges and molecular challenges that we'll see in much deeper content throughout the talk. So just to also sum up some of the management of sarcopenia, largely coming from the human field, well, the first obvious observation is there's really no approved drug in this field. There's been active research around it. There's been molecules that stimulate muscle mass that went up to phase 3 clinical trials. In most cases, they're pretty efficient at building back muscle, but what has been limiting is really the ability to regain functional muscle tissue, to really couple the growth of muscle to the contraction. And today, really, there's no approved drugs. The androgen replacement therapy does work, but has an unfavorable risk to benefit profile. So it's only restricted to really very rare cases. And by far, the best means to counteract sarcopenia is exercise. It's quite accepted, but the recent work has even shown that the type of exercise doesn't matter so much. What's really important is regular physical activity. And there's been two very large multicentric RCTs, one in Europe, one in the US, called Life and Sprint. We're talking trials that have over 1,000 patients here, and both have really shown that the best way to maintain active and functional muscle is to engage in the structured exercise program. And here we're really not talking about, acute training. It's really structured activity 3 times per week. And the quite interesting angle, I think, out of the straw that has been learned is there's the social component to this, because the groups that have been created to generate the intervention arms have also learned that putting the people together, that social interaction is very important. And I think this is where a point I want to make is I think with the relationship between pets and the pet parents, there's actually a unique opportunity to manage sarcopenia via this physical activity. Now, nutrition is, of course, central. This is one of our core beliefs. High-quality protein is important, especially because during aging, the amount of dietary protein that's required to stimulate protein synthesis increases. So there's really this anabolic threshold that is different in older people and needs higher content in branched-chain amino acid. And we'll see this comes with a few paradoxes in the context of aging. Slowly digestible carbs are important. You don't want to induce metabolic anabolic resistance, but on the other hand, and insulin resistance, sorry, but on the other hand, you do want to supply carbohydrates for energy. Micronutrients, we've heard about vitamin D. The literature is not fully consistent, but there is some support, at least in humans, that vitamin D does support muscle function. And the B vitamins are also quite important for energy. Finally, PUFAs, omega-3s, will support the management of inflammation. And there's emerging mechanisms, which I'll talk about today, on non-caloric parts of the diet, and especially the polyphenols for cellular mechanisms of aging. Now, aging is not actually linear, and especially in muscle, where we tend to think about the process of sarcopenia as 2 main phases. There's really a chronic phase that in humans occurs over decades, where weakness and inactivity goes hand in hand and slowly declines, and this happens over years. On the other hand, there's a much acute phase that happens when a traumatic event or a surgery happens. And there, because the surgery will lacerate some muscle beds or because the injury will cause damage, there's actually an acute drop that recovers owing to plasticity. But what we realize is the recovery is often never total, or at least is much slower than in the young age. And therefore, these acute events really contribute, and their accumulation contribute to this chronic cycle. So it's really 2 intertwined vicious cycles. And I'll talk a bit differently about these 2 aspects because we look at different mechanisms. So starting with acute management of muscle repair, there the core cell type that we're interested in is the satellite cell or the muscle stem cell. And these cells are very tiny cells. They're quiescent. They're basically inactive. They sit on top of fibers. They're very small compared to a fiber. And they wait for injury to happen. And whenever injury happens, there's actually necrosis, inflammation, and these signals that break their extracellular matrix activate them, and they start proliferating. Like any stem cell, they go through fate decisions to repopulate their own niche and maintain the pool. But the vast majority then proliferates and engages in myogenic differentiation to refuse together and rebuild muscle fibers. And you see that you can model this experimentally. It's actually extremely efficient. After a few days after an injury, you get no more muscle fibers. And within a couple days, the stem cells that are not affected by this injury, because they're so small and insulated, will then repair the damaged muscle. And nutritionally, there's really very little means to modulate the activity of these cells. So a couple years ago, we engaged into a phenotypic screen in the lab where we looked at some of the key markers of myogenic commitments so that we could boost their myogenic capacity. And we did this by treating human muscle stem cells in vitro and doing high-content imaging. And through this approach, I mean, really, we're talking about quite a wide screen of about 47,000 molecules. We really funneled it down to a few that had activity and a few that had confirmed activity and had a safe use. And the 2 best ones were pyridoxine and nicotinamide, so 2 micronutrients that as you'll see, stimulate these muscle stem cells. And what they do is shown in this slide. In a dose response, nicotinamide really boosts the amplification. So when the stem cells exit their quiescence, they start to proliferate. Well, there we boost that proliferation, and ultimately this leads to engagement into myogenesis. On the contrary, pyridoxine doesn't help at all proliferation, but it helps at the point where the cell has to exit again the cell cycle to re-engage into